Maybe you can start sharing your screen.
Okay. Do you want me to start into the presentation? Are we live now?
Yes, yes.
Oh, okay, cool. Awesome. Very good. So I should just, okay, let's,
Yeah, so we got really excited by your recent paper in PNAS,
yeah.
the phase separation in ER. And today we're really looking forward to your talk about this paper and more.
Okay, definitely.
I can see your screen.
Good.
Yep.
Very good. Okay, well, I want to thank you, Erdnak, for inviting me today. And I'm extremely excited to be sharing this work with everyone. Sorry, I can't make it. Actually, back here in the United States, most PIs like myself are actually homebound. And so this is my study right here where we're talking. Anyway, today I want to talk to you about biological roles of phase partitioning in membranes. This is an area that I think is extremely important in order to understand how intracellular movement between different organelles, as well as sorting of proteins in different environments within the cell, is taking place. I mean, we know a lot about cytoplasmic proteins like coat proteins that can bind onto the surface of membranes and sort molecules commensurately. But it's really been under, I think, it's only recently that we've really started realizing how important this sort of the biophysics of phase partitioning in membranes is playing a really important role in how proteins as well as membranes are being sorted within the cell. Now, this concept of lipid-based partitioning has been around for a very long time, but not sort of spanning throughout all of the cell biological realms, if you speak. Basically, in the 60s and 70s, people realized that membranes are made up of different types of lipids, cholesterol, sphingolipids, and just regular glycerophospholipids. I'm trying to see if you can see my pointer. Can you see my pointer? Okay, perfect. And they also realized that different combinations of these phospholipids can, in biological membranes, undergo phase partitioning into what are described as either liquid disordered, where the acyl groups are highly disorganized, allowing sort of for a more fluid type of membrane environment, to a liquid ordered environment where sphingolipids and cholesterol through hydrogen bonding networks can assemble into a more organized form, giving rise to a thicker, stiffer bilayer. And this was really shown, I think, pretty dramatically with giant unilateral vesicles, where if you mix these three different types of lipids and generate these giant vesicles, you can clearly see phase segregation of liquid ordered versus liquid disordered domains on the surface of these structures. Now, I would say probably around 10 years or so ago, people realized that you could generate the same type of phase partitioning on the plasma membrane by generating plasma membrane derived giant vesicles through hypotonic swelling. Ordinarily, if you look at the plasma membrane, it's very hard to see segregation of proteins into ordered or disordered domains, presumably because of the actin cytoskeleton and dynamic endocytosis and exocytosis that is recycling the membranes at that plasma membrane. It's hard to see this kind of spatial segregation of these different domains. And as a consequence, people in the field, at least of membrane trafficking that I'm very familiar with, were very skeptical of the significance of lipid-based phase partitioning for driving any kind of large-scale sorting events. But clearly, you can see that you can get very nice lipid phase partitioning in plasma membrane-derived vesicles, which have, the normal composition of lipids as well as proteins on that plasma membrane. And indeed, if you introduce different fluorescent proteins that are tagged to different types of proteins that, like transferrin receptor or GPI anchored proteins, which are known based on biophysical chemical assays to prefer either being in a liquid ordered or disordered domain, you can see by introducing these proteins into a living cell and then generating a giant millimolar vesicle, you can see beautiful segregation of the GPI anchored protein in green to domains that are depleted of the transferrin receptor in red here. And these two proteins are clearly sorting into the respective ordered versus disordered domains of this giant vesicle. So what I want to do with that background, today I'd like to talk about three different faces, if you will, of membrane partitioning and how we're realizing the fundamental role of this phenomenon in driving protein segregation in different cellular environments. So I'm going to start with ER membrane partitioning. And in particular, what we're seeing is there appears to be segregation of lipids at these organelle contact sites. The ER makes contacts with other organelles. And I'm going to show you some evidence that suggests that there is membrane partitioning at those sites. That is particularly surprising because the ER has very low levels of cholesterol and virtually no sphingolipids. So the fact that you can see partitioning in that environment, I think just underscores how fundamental the process of lipid partitioning is in all biological membranes. After talking briefly about that, I'm going to move into another area that we've been studying, which is lipid partitioning on the plasma membrane and its role in driving the budding of HIV viral particles. And then finally, I'm going to move back into the cell and look at membrane phase partitioning in the context of the vacuole, which is the lysosome in yeast. There's some really exciting results, I think, that are coming out from that area, and I'd like to share with you with that. So let's start with ER membrane partitioning at organelle contact sites. This project was really sort of a fun project initiated by a postdoc in the lab, Chris King. And basically, he really, as a PhD student, he had done a lot of cell swelling to look at giant plasma membrane derived vesicles. And since my lab does a lot of work intracellularly, in particular, we're fascinated with endoplasmic reticulum, he decided to he started looking inside cells that have been hypotonically swelled. And to his surprise and my surprise, what we found was that the endoplasmic reticulum undergoes a swelling into giant vesicles. These are giant intracellular vesicles. They're not blowing up. You can see that. So here's the what the endoplasmic reticulum normally looks in a control cell to the left here. And what you're looking at is a KDOL protein, which is a luminal marker here. And this is a ER membrane marker, Sec61. When we swell these cells, and by swelling, I mean, basically we're diluting the medium about 1 to 10, and that's sufficient to cause most of the cells to undergo this kind of gross swelling, where water is moving into all of these different organelles, swelling them out. What you can see is that the ER remains intact. You still see a membrane with Sec61. Inside that membrane is KDOL receptor, the luminal marker. You can see separation between these two different species, which is exciting because you can now start potentially looking at the way different proteins might be distributed within this organelle at a light level where you can distinguish different populations. Now, this swelling is reversible, and that's really important. These cells are not going off into a dead end. In fact, once you swell these cells, they are in some ways protected from death, if you will. When we bring the cells back to normal medium, we see the ER reacquire its highly reticular pattern. Oftentimes, this is followed by an apoptotic event 15 minutes later. And it's presumably because we've actually herniated mitochondria during the process of swelling. So this is all an aside, but it's a very interesting pathway. And the fact that we can see the ER reassemble this way is extremely exciting because it suggests that there's mechanism by which these membranes can actually refuse to retubulate the ER after there's been this kind of osmotic transformation and fragmentation of the ER in this way. Okay, well, given that we have the ability to visualize at the diffraction limited level how proteins are distributed on these big spherical vesicles, we decided to start looking at whether any two proteins on the ER might undergo some type of segregation based on lipid properties, phase partitioning properties. And in order to address that, we use the classic approach used in plasma membrane-derived vesicles to drive phase separation, and that is to reduce the temperature. So what you're looking at here is swollen ER vesicles in an intact cell where we've expressed a GPI-anchored protein, which we know will prefer a liquid order-like environment in the membrane that it's in. Sec61, which we know is probably not going to prefer being in a liquid ordered environment. And here the two merged. Now, as we drop the temperature, we saw something very, very interesting. The GPI-anchored protein started phase separating away from the Sec61 protein at these low temperatures, suggesting that the low temperature could drive phase separation within these vesicles. Now, the implication of this is that under normal conditions, the GPI-anchored protein is surrounding itself with very different types of lipids, and that those lipids are actually present in the endoplasmic reticulum at steady state, because there is no communication going on between these swollen ER vesicles. They are not budding things off. They are frozen vesicles sitting in a cytoplasm. And all we're doing is just dropping the temperature, and we see these proteins segregate. Well, in order to verify that what this represents is differences in ordered versus disordered character of the ER membrane, we use this push-pull pyrene dye, as shown here, which can be used to measure relative lipid ordering. And in particular, this dye undergoes a spectral shift to shorter wavelengths in ordered membranes. And what you can see here is that in the areas where Sec61 is depleted, which we think is an ordered region, that's where the GPI anchored protein would go, we see that there is higher relative ordering associated with that point. So we then started wondering whether the ER sustains its contacts with other organelles when we swell the ER in this manner. The ER is the largest organelle within the cell and makes contacts with virtually every subcellular membrane-bound organelle. And so we wanted, and those contacts are playing very important roles for lipid trafficking from ER to the organelle and vice versa. It's also extremely important for calcium and other metabolite exchange between organelles. So here we are looking at the ER swollen vesicles in the context of its association with plasma membrane, endosome, and mitochondria. And what you can see is that the ER is maintaining its contacts with all of these different organelles. Here's the ER making contact with plasma membrane. Here is it making contact with endosome. Here it's making contact, here's the ER making contact with mitochondria. Now, both endosomes and mitochondria swell under these osmotic conditions. What that means is water can move into these structures in response to our hypotonic swelling. Now, what's super interesting is that the contacts that we're seeing that the ER is making with organelles like plasma membrane here or lipid droplet here are sites where the membrane tends to be more liquid ordered like. And we know that because Sec61, as you can see here, is depleted from this contact region with the plasma membrane. And likewise, it's depleted from the contact region with the lipid droplet. We also see, and this is just showing it with a ER liquid ordered like probe, the GPI anchored molecule that we've trapped in the ER so that we can label the ER with this ordered probe. So here's your plasma membrane, here's the ordered ER probe, disordered ER probe, and you can see this kind of segregation. And it's very dramatic with the endosome contact. In red is the endosome. This pink here represents Sec61, and this is the GPI anchored protein that's retained within the ER. So you see a beautiful phase segregation at 7 degrees in the swollen cell system. So what that's suggesting is that these intraorganelle contact sites that the ER makes are sites of enrichment of lipids that would enable the contact site to have more liquid-like ordered characteristics. Now we also found, and this was done because we really wanted to prove that the sites where the ER is making contact with these other organelles, in fact, are organelle contact sites. And our evidence for that is that the tethers that facilitate these types of contact sites are localized at these co-associations of the ER with different organelles. And so here we're looking at mitofusin 2, which is well known as an ER mitochondrial tethering protein. And in red, here's your mitochondrial probe, here's our ER probe, and you can see that the mitofusin 2 is preferentially enriched at that junction, really confirming that when you see these contact sites, these are contact sites that are being maintained by these tethering molecules in response to this osmotic condition. So in summary, what this paper really offers is a sort of new way to look at how organelles can interact with each other through hypotonic swelling, which essentially by swelling these cells and the organelles within them, you just make things easier to be able to visualize the co-association of the organelles. And surprisingly, many of the tethering proteins that we've looked at still remain associated with the membranes and offer a very interesting strategy for then going in and understanding more about these ER organelle contact sites. And what our data is suggesting is that those contact sites are areas of preferential liquid ordered enrichment. Okay, so now I want to move on to the second topic that also relates to phase separation in biological membranes, and that is phase separation of lipids and proteins that are occurring during the HIV budding of the HIV virus. Now, HIV is one of several enveloped viruses, which also includes coronaviruses like COVID-19. These are all enveloped viruses, which means they're enclosed in lipid membrane. They're acquiring this membrane from their host. And what's important is that they don't acquire, it's not just a random association of the proteins from the host that gets put into these viruses. It's a unique protein composition. And so that raises the question of how are these proteins being selectively put into these viral envelopes, the membrane of these viruses. So as I said, we're going to focus on HIV. And one of the things that, and this is just a cartoon to the left of HIV and the way it buds. This is the GAG protein, which really forms the platform that is going to bud out and sequestered into this membrane surrounding that platform are proteins. Now, one of the things that we found is that all you need to do if you want to create this kind of viral particle is introduce GAG into cells. So to the right here, we have this is a movie sequence where we've introduced, I'm starting to play from the beginning, we've introduced 1 GFP GAG and 4 untagged GAG molecules. That's in order to allow GAG to properly form this tight scaffold. And that's sufficient to actually bud out particles from the plasma membrane of this cell that we've transfected with. It's pretty exciting. And if we go in and look at the plasma membrane of these cells that we've expressed the HIV GAG, GFP tagged, you can see, in this case it's flag tagged, you can see actual particles. This is correlative IPALM and scanning electron microscopy. You can see there's GAG in all of these particles that are budding off from this surface. So given that we have the ability to, at will, drive the budding of these enveloped particles, these HIV-like particles, we started interrogating how proteins get sorted into these structures. And I mentioned that it's a selective process, but many people in the field for many years have thought it was a passive sorting process. And so I'm going to take you through some data that really nails the evidence that this is a selective sorting mechanism. So here, what you're seeing is to the top is a palm image of envelope protein before we've transfected cells with GAG. You can see it's just distributed randomly on the plasma membrane. When we introduce GAG into this cell, you can see the envelope protein now starts undergoing clustering into viral-like particles. And this movie, which is a TERF, very fast time-lapse sequence of cells that are expressing GAG plus envelope protein, shows the co-recruitment of envelope into the GAG viral particle as it's emerging on the plasma membrane. Okay, well, that's cool. Well, are these viral particles specialized in their lipid composition? And for that, we looked to see whether they had enrichment in cholesterol or sphingolipids. In fact, prior work had suggested, based on purification of HIV viruses, that they were preferentially enriched in cholesterol and sphingolipids. And so we wanted to see during the viral budding process, can we see this enrichment in a living cell context? And that's what you're seeing here. We're introducing a GAG M cherry to the left here. And the next panel over is either a Bodipi cholesterol probe or an NBD sphingomyelin probe. And if you zoom in on areas where you can see GAG, you can see there's enrichment of cholesterol in these GAG punctate particle zones, as well as sphingomyelin. We also use the push-pull pyrene dye to show that the viral assembly sites are sites of enrichment in lipid ordering. So that was super exciting because it raised the possibility that the mechanism by which proteins are selectively sorted into the HIV viral particle is based on those proteins' preference for being in a liquid ordered environment. So we set up a bunch of experiments to try to test this specific hypothesis. What's the role of lipid-based phase partitioning in viral membrane formation? So we started by generating several lipid phase sensitive probes. And these are 3 molecules that we looked at. These 2 here, the PM-EGFP, has a myristate and palmitate residue that allow it to be anchored through lipid into the bilayer. This is a GPI anchored protein. Again, it's lipid anchored solely for its integration into the membrane. And then here's a journal EGFP, which should essentially prefer a disordered as opposed to liquid ordered domain. And consistent with that, when we introduce these lipid sensitive, phase sensitive probes into cells, these probes go to the cell surface. And then when we create giant plasma membrane vesicles, we can see that the general lipid anchored probe is excluded from ordered domains that are defined by cholera toxin, whereas the plasma membrane probe that's anchored by palmitate and myristate colocalizes with that cholera toxin distribution, as does the GPI anchored protein. So what do these probes do in the context of the HIV budding assay? If we introduce HIV GAG into our cells and start creating viral particles, will these lipid probes go into those particles? And the answer is the ordered probes do, but the disordered probes don't. And so here's imaging of the GPI anchored protein. You can see it's going right into the GAG punctate that defined the viral particle, as does the myristate palmitate probe that both show preference for ordered, being in ordered environments on the plasma membrane. By contrast, the journal probe, which does not show ordered phase preference, is excluded from the GAG enriched domains. So that was pretty cool. So that suggested, hey, yeah, maybe the way proteins are being sorted into the virus is through phase partitioning, the ability of a protein to prefer being in a liquid ordered environment. So that led us to say, okay, well, let's look at some authentic plasma membrane proteins that have been shown in prior work to prefer being either in liquid ordered or disordered domains. For instance, CD59, it's a GPI anchored protein. Envelope protein itself prefers being in a liquid ordered domain, presumably because it's got this palmitoyl probe moiety. And this GT46 is known to not want to be in an ordered environment. And you can see that because it's excluded from these toxin labeled membrane portions of the giant plasma membrane vesicles. Okay, so let's look at these probes in the context of a budding viral particle. And sure enough, only plasma membrane proteins favoring liquid order domains become enriched in the HIV membrane. And that's shown here. Here we've got, these are the GAG probes, and then here's CD59 enrichment in these GAG viral budding sites as the envelope protein enrichment. And we have either no enrichment or exclusion of this disorder preferring probe. So in order to sort of really further query this simple hypothesis of the way proteins get into this viral particle is simply by phase partitioning, we decided to test the effect of removing the envelope's palmitoyl residue. We just get rid of this palmitoyl residue. When we do that, we can see that this envelope protein no longer prefers being in the regions of these giant luminal vesicles where the envelope, collar toxin is located. So here's collar toxin in red, and you can see the envelope protein lacking that palmitoyl moiety is all that's needed to cause it to move out of that phase separated ordered domain. So what does this do in the context of the virus? Well, it prevents it from going into the viral membrane. So here we've introduced the palmitoyl deleted envelope protein into cells, and we're looking at whether or not it can co-cluster with the GAG enriched viral particles, and you see it does not, it's depleted. So thus the palmitoyl anchor is sufficient for mediating the MLV envelopes enrichment in the HIV membrane. Now, we've done a lot of work looking at a bunch of different proteins and how they sort. They all seem to follow this basic concept of if a protein prefers being in a liquid order domain, it ultimately is going to get into that viral particle. But what's interesting, and if a protein doesn't want to be in a liquid order domain, it will be excluded from the particle. And you can see the time course of this process. In fact, if we map out the rate at which proteins are being included versus depleted from the viral particle, you can see that different proteins are actually going into the particle site versus going out of that particle site at different times during the bud assembly. So basically we can monitor the bud assembly using the GAG GFP. So we have an assembly initiation time point, a completion time point. We can then, based on that sequence, we can place the co-association or the entry of these different proteins or exclusion of these different proteins into that viral site. So that's suggesting that as the HIV particle membrane is forming, it's actually, it's not a instantaneous process. There's actually things happening over the course of the 5 to 10 minute assembly process, where as proteins come in, other proteins go out, and this is the environment of that particle is constantly changing throughout that whole process. So that was exciting because it led us to start thinking about throughout this process, there being different things happening in that viral membrane that gives differential affinity for different proteins that want to be in an ordered domain. So it basically leads to and one of those questions that it leads to is how does the viral assembly site gain its partitioning capability across two membranes? So it's not only the inner membrane that is interacting with GAG, that sorts proteins. But the outer membrane also has changes in its lipid environment going on. So how does that happen? And we think it's through a type of transbilayer coupling mechanism. Basically, here is the structure of GAG, and it's got three different subdomains. This MA domain, which is called the matrix domain, is what interacts with membranes. And it interacts with membranes through a positive amino acid stretch that's highly basic. And we and others believe that that could essentially attract GAG to membranes that are preferentially enriched in either PIP2 or PS, which can bind to this highly basic amino acid stretch in this MA domain of GAG. And so the idea would be, and the reason why that's significant is because PIP2 is known to have very long acell change that can actually cross into the outer leaflet. And so does PS. And so the idea is if you start coalescing, accumulating PIP2 and PS on the inner leaflet, those acell chains will essentially start impacting the outer leaflet and start drawing in other long chain acyl chain, either proteins or lipids like sphingolipids and GPI anchored proteins. And so the idea would be that this transbilayer coupling driven by GAG's interaction with PIP2 and PS would enable this whole membrane to start becoming much more ordered because you'd be drawing in sphingolipids and cholesterol. So how do we test this transbilayer coupling model? Well, the way we've done it is to get rid of the MA domain on GAG. That MA domain has that positive charge stretch that we think is critical for recruiting to binding to PIP2. So here's the molecule we created. It's a GAG that doesn't have the MA domain. Instead, we've replaced this portion of GAG with a sequence that allows myristoyl and palmitoyl to be added to GAG. And through the myristoyl and palmitoyl groups, the GAG is able to bind to membranes. The question is, it can bind to membranes and GAG itself can multimerize. Is this sufficient to create a phase separated domain, a liquid ordered domain that would recruit things like CD59, which is a GPI anchored protein that doesn't cross into the inner leaflet. It's only an outer leaflet anchored molecule. And so what we found, amazingly, is that CD59 does not go into these GAG delta MA clusters. So we can get GAG to cluster when we remove this MA just based on GAG's affinity for itself. But those clusters do not recruit these ordered proteins, CD59. And this is in contrast to the wild type GAG, where you can see very quick recruitment of CD59 into these domains. So here we've got a comparison of the GAG without the MA domain, where you do not see a CD59 going into the particles that are forming under these conditions. By contrast, the wild type GAG that is essentially driving transbilayer coupling is driving the sorting of CD59 into this structure. So basically, we think that this type of transbilayer coupling is playing an important role in how you generate phase separation at that HIB assembly site. Now, there's one other piece of this puzzle that we think is going to be important, and that has to do with membrane curvature. So it turns out that the HIV particle is a highly curved particle. The GAG has an inherent curvature that leads to budding out of the viral membrane platform. Now we know that when lipids, liquid ordered lipids and disordered lipids phase separate, you have what's called line tension segregating these different domains. That line tension actually is lowered if the membrane buds out, because the diameter of that line tension is reduced. So curving the assembly site decreases the line tension, which we think is enhancing phase separation and efficient protein sorting. Well, how can we prove that? Well, what we've done is generated a GAG-like molecule that has the MA domain for partitioning, for transbilayer coupling, but it lacks the rest of the GAG moiety that inputs curvature to the oligomerized structure. So the CA domain of GAG, as it multimerizes, actually puts a curvature to the membrane that is associated with. So what we did is we got rid of this CA domain, so there's no curvature, and we replaced it instead with proteins that don't curve, essentially a EGFP that has EGFPs tagged to a dimerizing protein. Basically, we tag this dimerizing protein to the MA domain, then add the dimerizer to force these molecules to aggregate, to come together. And now we're going to ask, do we get phase partitioning of proteins when you don't have curvature? Because this thing does not curve. And what's incredible is you don't get CD59 sorting under these conditions. So here you can see that this artificial dimerizing, aggregating, oligomerizing protein forms structures on the plasma membrane. Those structures are not, they're aggregated proteins. They're not curved. They're not creating a curved structure. And you see that CD59 does not go in there, even though you have everything ready for transbilayer coupling. That's not sufficient to hold CD59 in that domain. And that's in contrast to the wild type GAG that has both the curvature associated with it, as well as the transbilayer coupling. So, and this is just the analysis of that. So basically our model for lipid and protein segregation into forming HIV particles is one where you have a scaffold, GAG, that undergoes oligomerization. GAG's ability to interact with PIP2 as well as phosphatidylserine enables it to create a transbilayer couple, if you will, that in principle will allow proteins on the outer surface, like GPI anchored proteins, to partition into this liquid ordered environment that it's creating. But curvature offers an additional attribute to this system. That curvature and the reduction of the line tension allows this viral protein, the viral envelope, the membrane, to capture, to hold these proteins that otherwise would just diffuse out. The partitioning is not great enough unless you introduce this type of high curvature. So anyway, it's really fun to see how all of these characteristics are playing a role in this process. Now, I want to, in the next 5 minutes to the end of the talk, go into one final area of the cell where phase separation is playing a really important role. And this role is not just sorting proteins. This role is actually driving a whole physiological state. it's in yeast, and we're going to be looking at the vacuole membrane. Well, it's been recently shown by Prince, other people, as well as ourselves, that the vacuole surface will remodel into this soccer ball-like pattern in response to starvation, where you have discontinuous liquid order domains that are surrounded by continuous discontinuous order domains surrounded by continuous, got mixed, continuous disorder domains. Now, this happens when cells undergo acute glucose restriction, or as cells are entering their stationary phase, and these are yeast cells. These domains that we see are truly phase-separated domains. They're highly dynamic. They hold proteins that prefer being in ordered versus disordered domain. And this is just a movie showing that type of dynamism. These domains also are enriched, the ordered domains, that is, are enriched in PI3P. And that you can see here with this GFP5 domain probe that's enriched in the ordered domains, the discontinuous ordered domains under glucose starvation. So the question is, how do these domains arise? And what we found is the domains require sterol. If we deplete sterol using a TET on-off system, controlling ergosterol biosynthesis in yeast, as we drop the concentration of ergosterol, we lose the microdomains on these vacuoles under glucose starvation. So that raises the question of where does the sterol and sphingolipid come from? We've found that it's coming from macroautophagy that is being induced under starvation. So the vacuole is a lysosome in yeast. And what happens under acute glucose restriction, as well as under rapamycin treatment, both conditions that are nutrient stressors within cells, you see increased macroautophagy. And this macroautophagy we found is absolutely critical for the formation of these partition microdomains. In fact, if we introduce mutations, deletions of key autophagy components, HEG6, HEG8, or HEG14, we lose the ability to phase separate the vacuole under starvation conditions. Okay, so what do these vacuole domains do? That's the $100 question. And what's really amazing is they're segregating proteins, not surprisingly, but it's amazing to see this in, these are freeze-etch images from John Heuser, where you can very nicely see the liquid ordered and the disordered domains on the surface of these glucose depleted cell vacuoles. So here's the vacuole under starvation, here's in fed. What you're looking at here is the vacuole ATPase, VPH1. And you can see that the vacuole ATPase is actually moving into these liquid disordered domains that are surrounding these relatively smooth area, suggesting protein depletion in this area. You can see these particles by freeze fracture. Those particles are proteins that have large domains, extra cytoplasmic domains. Vacuole ATPase, no doubt, is one of them because we see that vacuole ATPase in the disordered domains. Okay, well, this type of segregation of these proteins might play a role in activating these proteins. And we were interested in that, in particular, the vacuole ATPase. Does it change in any way if we drive phase separated domains? And Evidence suggesting that is shown here, where we're looking at the ability of that vacuole ATPase to pump protons into the vacuole to make it more acidic. And this pink shows enrichment of this acid probe, quinacrine, in the vacuole of a yeast undergoing 21 hours of acute glucose restriction, where there's no glucose in the medium at this point in time. And you can see that these cells are just pumping protons. I mean, this is in a highly acidic vacuole. In contrast, if the cell has not been acutely glucose starved, or if it's only been gradually glucose starved, you see there is no enrichment. The pH of that vacuole is not highly acidic the way it is under acute glucose restriction. And here's where we've disrupted the ability of the vacuole to phase partition, either with the autophagy mutant HEG14 or HEG6. And both these mutants prevent the vacuole from being able to acidify. Well, why is it important for the vacuole to acidify under starvation? Well, one thing is that the vacuole is playing a critical role in recycling proteins and lipids. And consistent with that, we can see that lipid droplets are actually being taken up into the vacuole under glucose starvation. And what's amazing is that the lipid droplets are actually being taken up via these, by docking onto the liquid order domains on the surface of the vacuole. So here's the vacuole that's showing its partition domain because the cell is starved of glucose. This is a lipid droplet marker, and you can see that the lipid droplet marker is docking onto these liquid ordered domains and then being internalized. So if you do time-lapse imaging, you can see the lipid droplet in red being drawn into the luminal environment of that vacuole. And this is just time-lapse imaging showing that process. So here, this is a lipid droplet inside the vacuole that's been taken up by associating with this smooth liquid ordered domain that is then internalized. Now we think that the fact that the vacuole is highly acidic, it's got a very low pH under this acute glucose restriction regime, is important to enable the cell to digest those lipid droplets. So here's our model in terms of what these partition domains are doing. Not only are they triggering the sorting of the vacuole ATPase to a domain we think where it's more active, creating a more acidic luminal environment of the vacuole. But these domains are sites of docking of lipid droplets, which then get internalized through a type of microautophagy. And the low pH enables those lipid droplets to be broken up into fatty acids, digested into fatty acids. And these fatty acids are going to serve as the fuel for long-term survival under starvation condition. So my last slide sort of summarizes this, as well as some other interesting material that I haven't had time to talk about. It would take too much time, but I want to share with you because I think it's really amazing in terms of how these partition domains are involved not only in sorting proteins to get the vacuole active, taking up lipid droplets, but we know that these partition domains are sites of organelle-organelle contact. So the ER vacuole contact occurs at these ordered domains, and we think is a conveyor, is a site where lipids can be conveyed back into the ER. We've got tons of lipids coming in through autophagy into this vacuole. At some point, it's going to get huge. What muffles that is lipids moving out of the vacuole back into the ER. And this plays an important role because it's the ER that makes lipid droplets. And if the lipid droplets are ultimately going to be delivered to the vacuole to create the energy that the cell's going to use you got to keep making lipid droplets. So this is a pipeline where macrotophagy is delivering lipids from the cell into the vacuole. Those lipids partition, and now you have a pipeline to funnel some of these lipids back into the ER to make more lipid droplets, to allow the cell to begin to eat these lipid droplets, delivering the these high carbon chain components to the mitochondria. Now, one of the things that we found is mitochondria actually proliferate under acute glucose restriction. That allows these cells to actually live for days without food. And that proliferation is also dependent on this lipid pipeline where lipids are being carried from the vacuole back into the ER and then either put into lipid droplets or through ER mitochondrial contacts delivered into the mitochondria, so the mitochondria can keep functioning under these conditions. When there's no glucose, there's no glycolysis. So the only way the cell can make ATP is through oxidative phosphorylation in that mitochondria. And that oxidative phosphorylation requires these fatty acids to be released from the vacuole. So with that, I want to end and thank you for your attention. The key people who did the work that I talked today are listed here. Chris King did the ER phase partitioning work. Prabh and Gupta did the work on the HIV budding particle. Chris O'Bara was involved in ER, some of the ER work, and Arnold Seo did all of the work connected to the vacuole in yeast. I also want to thank our collaborators, Greg Voth, Mark Johnson, collaborating on the HIV work, as well as Jean-Baptiste Masson and Florian Scarlata and Sepp Colvane for help with the work in the yeast vacuole. Thank you.
Thank you very much, Jennifer. Amazing. It's really a fascinating work. So we have a lot of questions. I hope you have a bit of time to answer the questions.
Yeah.
And the questions won't be chronological because they were kind of mixed questions for different sections.
Okay.
But I will start chronologically.
Okay.
So the first question is by Sarah Keller, and she's asking, from Chris King's work, what is the scale, the diameter of the giant ER vesicles? So what would be the average of these ER vesicles that you get?
I'd say about 5 microns or so, 2 to 5 microns. I mean, it varies dramatically.
And for mitochondria, is it also similar?
It's a little bit smaller, but what's really interesting with the mitochondria is the matrix actually swells more than the intermitochondrial region. And so that's where you get this, this type of herniation of the mitochondria. And because basically the matrix busts slightly out of, we can see areas where mitochondrial matrix has swollen out from the outer membrane. And that, we think, is how these cells die when we bring them back into normal medium. It takes about 15 minutes, but they will die. Because any cell that has a herniated mitochondria, that's apoptosis is triggered, and that's the end of them.
Thank you, Jennifer. The second question is by Mark Tisopikul, and he's asking, over what time scale does the phase separation occur upon cooling?
Pretty quick. I mean, you know, within minutes or so. Yeah, I mean, it doesn't take hours.
Okay.
It's not like, it's we haven't sort of done serious time-lapse imaging of that process. I think if you really want to get the clear-cut answer to that question, an email to Chris King would do the job, because he's the one who's done that time-lapse, has looked at it in much more detail than I have.
Thank you, Jennifer. So the next questions are by Ganesh Shelke. She says, I'm wondering if the plasma membrane vesicles in your experiment system derived from hypotonic swelling is different from microvesicles derived from membrane vesicles.
Absolutely. The microvesicles are like HIV particles. So if you're interested in microvesicles, you're interested in the second part of the talk. It's very similar, we think, to the way HIV is budding out from the plasma membrane. Those are, we think, and actually there's a lot of GAG proteins that are in the genome that we think are actually doing exactly this to create microvesicles or exosomes.
In
So it's
Ganesh's,
not like the GUVs at all.
in Ganesh's next question is, does that affect the partition kinetics in the systems?
Does what affect the
difference between probably the vesicles, because it's the second part of the question, yeah.
Oh. Yeah, I mean, the giant, the plasma membrane derived giant vesicles are just a, they're just passive representation of the plasma membrane. There's not sorting into those structures, as far as I know. Sarah may have more insight into that, Sarah Keller, who's on this call. But my understanding is that the plasma membrane derived giant vesicles are just basically part of the plasma membrane that's swelling out because you're depolymerizing actin and you get an osmotic imbalance as a consequence. Budding of the HIV virion is a very choreographed process that requires the GAG protein that's oligomerizing and creating a specialized environment in the membrane that it's associating with.
Okay, the next question. When you hypothetically swell the cells, does all organelles get equally swollen? If not, then will this affect the lipid exchange protein interactions at the membrane contact sites?
Yeah, so there are certain organelles that don't swell. A lipid droplet doesn't swell, and peroxisomes don't swell, which is sort of interesting. And I think it says something about the contents of those organelles. I mean, a lipid droplet is just a monolayer lipid with triacyl glycerides and cholesterol esters. There's no way it's an osmotic effect when these cells swell. There's water input into these organelles. And so it's understandable why the lipid droplet doesn't swell, because it doesn't have water. But peroxisomes, the fact that they're not swelling, suggests something very interesting about the luminal contents of a peroxisome. The other question is, do all the organelles swell equivalently? There is heterogeneity. I mean, if you go into our PNAS paper, what you find is that not all cells will swell at the same time. Some cells, you see beautiful uniform swelling, and others you don't. We don't understand what explains that. I mean, it's a very crude mechanism that we're using for swelling these cells. It's clear that further work in this area will potentially help us understand how to make this more routine, so to speak, to answer some of these questions.
Thank you, Jennifer. So the next question you answered, I guess. If you do not provide fresh medium again, how long do these vesicles sustain until the cell undergoes either apoptosis or necrosis?
Yeah, so they stay forever. I mean, it's amazing. They're protected from apoptosis. So it's only when you bring them back down to normal isotonic medium that they will die. So it's one way you want to keep a cell alive, swell it like that.
Yeah, it's not a happy life, but it's alive.
No, it's not a happy life. But the thing that's really interesting is, when we first started these experiments, people were saying, well, it's so unphysiological. But there's lots of environments within, animals where cells are experiencing dramatic changes in osmolarity. And even, these blebbing motility, when you look at organelles in that blebbed area where the cells are undergoing this blebbing motility, those organelles are essentially fragmented and swollen, very similar to what we're seeing in these osmotically swollen cells. So I think this is really, we're just sort of at the tip of the iceberg in terms of understanding how these organelles are responding to these different osmotic challenges. In fact, one of the things we're trying to look at right now is whether under glucose starvation, this beautiful vacuole that you're seeing here, is that swollen or not? What's the osmotic pressure? Is the swelling itself part of the mechanism for driving some of this partitioning that we're seeing?
Thank you very much, Jennifer. Next question is by Nidongo Lee, and saying in the contact with organelles, there should be many protein tethers that would exclude Sec61. If Sec61 is excluded from the contacts, maybe it's just due to exclusion effect, but not the domains.
Agreed. That's true. Yeah, we don't know how it's excluded. We do know that that domain has enrichment in ordered lipids based on our push-pull pyrene dye localization, which is it's sensing lipid order. So we know it's ordered, but whether that ordering is sufficient to exclude Sec61, I agree. We don't know. I mean, in the case of HIV, we showed that without curvature, we didn't get sorting of certain proteins, even though we know that you can create a type of low-level enrichment of liquid ordered proteins in that domain. So I agree with what you're saying.
But also, you also showed that the GPI can go in it, right?
Yeah.
So if it was super packed and it was excluding anything rather than this binding partners, it would also exclude.
Yeah. So GPI could go into it. Yeah, you're right.
So next question is by Yin Dong. Does the LO still congregate at the contact size if swollen at 37 degrees?
Does the mitochondria?
No, LO domain.
Oh, still go into the contact site at 37 degrees? We don't know. Because we don't see, you're only, it's only when we lower the temperature that we're seeing the phase separation of these proteins and the lipids. That doesn't say that those environments, the contact sites, don't have a enrichment or a sort of a dynamic type of enrichment in terms of these lipids, but it says that they do not become, they don't go into a dramatic form, where they have a visible character, unless we go down to that low temperature. And that's the same with giant and normal vesicles as well as plasma membrane derived vesicles. You need to drop the temperature in order to see that phase separation because it drives it.
Thanks, Jennifer. Next question is by Jacob Koźnacki. And he says, we have done a similar study using scanning STED FCS. And in our hands, we did not see any enrichment of sphingomyelin in the budding sites. Any thoughts why this discrepancy would be?
I guess, okay, the question would be, how are you creating the budding site? I mean, what time point are you looking at that budding site in your FCS movies? I don't know, how else to explain that discrepancy.
Probably Jacob can get in contact with the preps.
Yeah, no, that would be really good. That would be great. Yeah, I'd like to get in contact with that because, yeah, it would be interesting to know why the FCS isn't detecting it. Mm-hmm. The question is, do you detect cholesterol? That would be,
Okay, thank you very much. So next question is by Chase Amos. Do GUVs formed with ER mimicking lipid composition exhibit the phase separation that is similar to the ER vesicles?
Repeat that. Do the
GUVs formed with the ER mimicking lipids, ER
Oh, okay.
lipids isolated from ER is similar to the ER vesicles that you obtain by swelling.
Yeah, so we haven't done that experiment. We haven't purified ER membranes and made GUVs to test that.
That would be actually interesting because, as you said, sphingomyelin and cholesterol, it's very, very low. I don't know if ER lipids would still phase separate.
No, exactly. And there's no sphingolipids in the ER. So the cholesterol is the sole sort of organizer. And yeah, and it could be that that organization really depends on contact sites with other organelles. The tethering proteins are participating in that, in which case, if you just made ER, if you purified ER membranes and then tried to do these experiments with ER-derived GVs, you wouldn't see this. But it's worth it doing. I mean, that's a really cool experiment to try.
Thank you, Jennifer. Next question. Shinzhou asking, does lipid phase separation happen on ER membrane under physiological condition? I'm sure you get this question a lot. And if so, is it possible to use any probes to observe it?
Does lipid phase separation occur on the ER membrane under any conditions?
Physiological conditions.
Yeah. Well,
and if there is any probe to observe it.
Okay, so we have another study where we've been looking at ER exit sites, which are sites where proteins leave the ER through COP2 vesicle budding. And we found that those sites are actually cholesterol enriched, and we use Bodip cholesterol to look at that. So that would be an example where there is cholesterol enrichment at an ER exit site. In some respects, it makes sense that that would be the case because the proteins that leave the ER all have long transmembrane domains, and many of them are actually oligomerized. So cholesterol would be extremely valuable in terms of stabilizing that environment. And sure enough, we see that. So, you know, if you want to see spatial segregation, look at ER exit sites. But I should emphasize that COP2 and COP1, which are at these ER exit sites, COP2 in particular, is dissociated under this. When you swell these cells, these cytoplasmic coproteins go off and stay in the cytoplasm. So you will not be recapitulating budding of vesicles out of the ER under these conditions, because these coproteins you don't have the conditions under osmotic stress to allow their recruitment.
Okay, so a related question by Maria Livia Sassano. Have you tested if the property of the membrane's contact sites are affected in swelling conditions? For example, the lipid trafficking?
No, we have not. No. That's hard. I mean, it's hard to see lipid trafficking anyway, even in a living intact cell, you know, to really monitor it. So to do that in one of these swollen cells could be super challenging, but it's a good question, definitely.
Okay, next question is by Pablo, and he's asking, thank you for your beautiful work. Is phase separation only observed under stress disease conditions, as you showed by HIV infection, starvation? How do you imagine in the PM in the resting conditions?
Okay, fair enough. Well, endocytosis, you know, is an ongoing process. I think there's good reason to think that there could be some phase separation happening at these sites of endocytic uptake. We haven't looked at that, but the fact that the principles that we're realizing that use phase partitioning for these remodeling events of membranes would point to a real possibility that there's some sorting going on of lipids during endocytic uptake. You know, I think that's an area that you could really try to investigate. It's challenging, you know, these experiments. I think part of the reason why people have been very hesitant to appreciate the role of phase partitioning in physiological contexts is that it's hard to see. You know, we use diffraction limited imaging, and these membranes are highly dynamic, and you know, the phase partition domains, some of them are very small. In the case of HIV, it's really small. But because we have, we know exactly where that HIV particle is that's forming, we have turf microscopy to allow us to just look at the plasma membrane. We now can see these things. I would not, definitely cells, this seems to be an important, phase separation seems to be a very important process under these perturbant conditions, osmotic changes, infection and viral infection, and starvation. But this is physiology. I mean, that's what happens to all cells. A large chunk of the human population right now is infected with a particular virus that is keeping me at home right now. I'd like to understand how that virus is actually working inside of a cell. And if it involves these principles, this is really important for us trying to figure out how to combat that virus. The same is true for starvation. Every time you go to bed at night, your body's going through a starvation phase, a fasting phase. And this is part of the physiology, deeply integrated into the evolutionary history of eukaryotic cells. So yeah, I'm really passionate about phase partitioning, as many of you probably are too. Otherwise, we wouldn't be having this session. So
yeah, true.
Yeah.
And I think what was fascinating, especially about the virus work you showed, you had electrostatic interactions, you had lipid sorting, and then the curvature. All these biophysical properties of the membrane are basically playing role to have one single outcome, which is the virus budding.
Yeah.
Right.
Yeah.
So it's not really an isolated thing that you have only lipid sorting.
And the thing that's really interesting with HIV virus is that what people don't understand, I mean, one of the reasons why it's been so hard to have a vaccine against HIV is because it has so few envelope proteins on its surface. They're separated far enough away from each other that antibodies can't cross-link them. And as a consequence, you can't create an immune response. And because of that, HIV is, we can't cure it. We can't generate any kind of vaccine, et cetera, against it. So the question is, how do you get so few envelope proteins on that membrane to sustain this? And we think it's tuning of these phase partitioning characteristics that HIV virus has done in order, if the envelope protein's partitioning in and out of this environment with the curvature cinching it. Basically when you drive that curvature, you stop that partitioning. It's all a balance of who comes in early versus who comes in late when you drive that high curvature to stop the partitioning. So we think this is going to be really, really important in understanding how viruses really fine-tune their protein composition on their membrane.
Yeah, I think it's also a very good example that phase separation is not only seeing a green and red domains, it's physiologically relevant.
Oh yeah, yeah, yep.
So one question related to this, you created this MA mutant.
Yes.
Does it bud from the cells?
No.
So it doesn't bud. So it assembles, but it doesn't bud.
The MA mutant that
doesn't interact with the PIPs and PS.
Right. It will. It does not bud. It just forms a platform.
It's very interesting.
Right. And the other one also, just when we do just the dimerization, it forms a platform, but has no curvature.
Brilliant. So, okay, next question by the audience is by Marina Garcia Macia. This kind of lipophagy is microautophagy specific for lipid droplets?
Yes. Under acute glucose restriction, yes. But we think that this microautophagy is probably playing a huge role in other turnover of other things from the cytoplasm, aggregated proteins, for instance. RNA particles could be turned over this way, where it's just direct invagination. But under acute glucose restriction, those phase partition domains attract the lipid droplets, and lipid droplets go in. It's just unbelievable. I mean, it's so exciting. I love it.
Actually, a related question, again by Xin Zhao. What is the molecular mechanism governing the specific lipid droplet docking at the Vacuole LO region? Any proteins involved?
Yes, ATG6 and ATG14. If you look in our paper, 2017 eLife, CO et al., it discusses the proteins that are required for that.
Okay, another question by Yongin Wang, and asking, do you have any idea why paroxysms cannot swell?
No. I mean, it's something, I think it has something to do with what's in them. You know, the paroxysms, you know, they're involved in actually lipid beta oxidation of long chain fatty acids. They're actually making precursors for myelin that's important in the brain. So they're probably lipid rich, but we don't know. I mean, at least I don't know. If you talk to a peroxisome expert, they probably could give you a good answer. But I was excited to see that because I just hadn't been thinking about peroxisomes as just sort of a solid sort of lipid kind of thing that wouldn't swell.
And also another question saying that what happens to Golgi when you swell in the Golgi, if the ER-Golgi contacts are still preserved?
We wanted to look at that. The Golgi, we couldn't, it just sort of, it didn't sustain itself very well. It was hard to see what happened to the Golgi.
Okay, so next question is by Daniel Tversky. Amazing work. Related to this is the work of Kaufmann, Schneider, and many others that suggest cells could control membrane phase state for communication, such as transmission of the action potential. It's more like a comment than a question.
Oh, wow. I haven't thought about that. That's cool.
And next question is Devrimers. How does lipidation of ATG8 affect vacuolar domains under starvation condition?
Lipidation of
PE. How is lipidation PE of ATG8?
We need, if you do not have, if we knock out ATG8, you don't get phase partitioned structures because we think you cannot deliver, you don't have macroautophagy going on. Without macroautophagy, you can't deliver membranes to the vacuole, to allow that vacuole to grow, to become enriched in sterols that drive the phase separation. And I didn't have time to go into this, but we've looked carefully at the type of macroautophagy that is involved under acute glucose restriction that we think is important for delivering the sterol. And that macroautophagy we think is from some sort of endosomal compartment, which would be sterol enriched, sterol and sphingolipid enriched, which would fit with the idea that it's macroautophagy initiated by ATG8 and all of its cohorts that's really delivering these lipids to the vacuole for phase separation. And those lipids ultimately are going to be taken back into the ER to allow lipid droplets to form and to allow the mitochondria to proliferate under starvation. So it's a whole lipid pipeline that goes into action under starvation conditions.
Thank you very much. Would you expect that if replacing the MA domains on GAG with LAC2 domains, which can still bind PS in the inner leaflet, rough domains will be induced?
Yeah, you get RAF domains, but they're not going to do, you don't get transbilayer, well, you won't get GPI anchored protein clustering because you won't get transbilayer coupling. But that's a really good point, and probably we need to demonstrate that.
Thank you, Jennifer. Okay, next question is by Martina Lukushevich from Oxford, and she's asking, would it be possible to force selected proteins to the HIV viral particles, for instance, via addition of palmitoyl residues, in order to make viruses more susceptible for the immune system recognition?
Definitely. Absolutely.
Okay, so we are almost through. The last question is Hongyin Wang again. Did you detect whether ordered domains can be coupled to other organelles that make contact with the enlarged ER through the ER? Oh.
Yeah, we've seen three-way coupling of organelles with the ER. In the paper by King et al., we can show that peroxisomes are in contact, peroxisomes make contact with the ER at the interface between the ordered and disordered domains. And then if you visualize where endosomes are, you see that endosomes are at those ordered-like domains, ER ordered-like domains. I want to say ordered-like because we don't know exactly what kind of lipids are in there, other than presumably cholesterol. But yeah, no, it's, I think what's cool about the ER partitioning stuff is it just starts you thinking about the logic in this that the cell has for how it's communicating all of its organelles through these contact sites. The fact that they can be preserved under a condition like osmotic swelling, where you can actually visualize pretty easily these contact sites, just opens up all kinds of possibilities for testing different hypotheses about these contact sites.
This is beautiful. Thank you very much, Jennifer. That was a very, very big pleasure for me, and everyone enjoyed it.
Yeah, well, thank you so much. I really appreciate it. Yeah, and hopefully the next time we all meet, it will be in person.
Yeah, exactly.
Really fun.
I hope your findings in virus will accelerate.
Oh yeah, definitely. Anybody out there, yeah, this is where you should go, is we need to figure out this coronavirus and the way it's functioning in cells. And there's a lot of cell biology that is totally not understood, but it's an envelope virus. So there's a lot that's shared with, what we're seeing with HIV at some level. So anyway, good luck to everybody. And thank you so much for listening to me. And I'm happy to address, if you have any questions or anything, just email me.
Thank you very much. Thanks, Jennifer. Great cell biology, great biophysics, amazing combination. Thanks a lot.
Yeah, thank you so much, Erdnik. Yeah.
Bye.
Bye.